We always wonder how birds can fly in many different ways. This is because of their flight feathers. We want to learn how flight feathers are made so we can understand nature better and learn bioarchitecture principles. Therefore, we organize a multidisciplinary team to study. The flight feather is made of two modules, the central shaft and the peripheral band. Both are produced by stem cells in the follicle under the skin. Here we will show the biophysical study of the rockies, molecular study of the barb, and the amber fossils that record the evolution of feather vein.
When birds fly, the strong but light central shaft of a feather, the rachis, supports the feather to work against the mechanical loads from air. The rachis is composed of the outer rigid cortex and an inner porous medulla. Resolving the detailed integration of these two components reveal the architectural principle of the diverse feather shaft in nature. We developed a novel analysis, QMorph, to resolve the cellular organization in the medulla. After coarse-grained measurement of the cellular size, orientation, and elongation over the entire rachis cross-section, the spatial distribution of the cellular morphology in medulla is quantitatively revealed.
We discovered that the rachis is integrated quite differently in nature.
Combining the QMorph analysis of medulla with the geometrical and material characterizations of cortex, we found that the rachis composite is integrated ingeniously with a simple and light medulla core with a polarized cortex reinforcement in the feather of good flyers. The water diving penguin even shows the extreme reinforcement of rachis, the cortex-only configuration. While the deformable modulary cells are the traces of the complex rachis development, the variation of QMorph patterns suggests an evolution of the keratin allocation strategy in the feather rachis to overcome the ecological challenges. We further studied the molecular control of cortex and medulla formation in the feather rachis and found BMP signaling can control the cortex thickness, while TGF-beta signaling enhances medulla formation.
Here is the contour feather shows two types of vein. Vein is composed of barbs. Each barb branches into barbulus. In one proximal pulmonaceous feather, barbula has no structure. In distal pulmonaceous feather, specialized ectodermal structure called hooklet is formed at the end of the barbulus. Hooklets hold barbs tightly together, providing the integrated structure of the feather vein for flying. But how do hooklets form? Dr. Weilin Zhang decided to take a look at how hooklets form at the cellular and the molecular level. In one contour feather, vein formed from distal pulmonaceous to proximal pulmonaceous feather. All these processes happen in the same dermal papilla in the bottom of the follicle. Our laboratory studies show that dermal papilla contains spatial information controlling pulmonaceous versus pulmonaceous branching patterning. To investigate the cellular and molecular mechanism, we collect follicles at appropriate time as they form. First, we dissect the dermal papilla for transcriptome analysis to find out differently expressed genes. Further confirmation is go through biological studies and functional assay in chicken. On the other part, we examine the barb ridge to understand the cellular morphology and the molecular expression under microscopy. Our result shows that asymmetric wind signaling within the dermal papilla affects the molecular expression and control barbular cell phase for branching formation. Functional studies done in the chicken shows that wind signaling modulates feather branch type formation. Increased wind signaling in the anterior of the dermal papilla, a pulmonaceous-specific hooklet-like structure formed in the growing pulmonaceous feather follicle. Then, how is the feather in dinosaur?
So Cretaceous feather embedded in Burmese amber have no barbule cell shapes with no shape difference between proximal and distal barbules, and there is no sign of hooklet. This indicates early feather veins formed through the overlap of proximal and distal barbules, and a modern feather veins configuration formed by hooklet, which may have evolved later.
Thus, we learn how simple epidermis is transformed into flight feather. Rachis become complex in feather dinosaur. but become simplified in different ways in current births. Barbs changed from radial to bilateral symmetry to bilateral asymmetry at the ramus and the barbule level, creating many functional forms. Together, they power birds into diverse ecological space.
At the application level, our multidisciplinary approach is capable of resolving various details of cellular compositions in tissues. The cortex medulla composition with different organizations of cellular origami makes the rachis a shaft with tunable material properties. The vane is made of fracture-like paper cutting principles with highly effective branch interweave. These modular enhance the functional flexibility of a fly feather. The architecture principles we have learned from nature in this study may inspire the development of novel composite materials and promote the advance of additive manufacturing technologies.